So thank you everyone for joining us. We'll be getting started in a moment. Before we begin, I'd like to share some important housekeeping notes. Please note that we are recording this event. We are also keeping the audience muted to minimize background noise. And please use the chat feature to pose questions to our speaker. Now it's my pleasure to introduce Tina Chung, President of the Prytanean Alumni, Inc., the alumni chapter of Prytanean Honor Society. Welcome.
Thank you. My name is Tina Chung, preferred pronoun she, her, and I'm the president of Prytanean Alumni, Inc., the alumni chapter to the Prytanean Honor Society. Prytanean Honor Society was the first women's honors organization in the United States, founded on the Berkeley campus in September 1901. The three women founders had a keen interest in student healthcare and housing issues, and the organization's work concentrated on these areas for many years. In later years, our work expanded to include awards and support for outstanding scholarship and volunteer activities on behalf of the university by students, faculty, and campus organizations. This work would have not been possible without the generosity of past, current, and future alumni, volunteers, and donors. From us to all of you, thank you, thank you, thank you. And to learn more about us, please visit alumni.berkeley.edu/Prytanean, Prytanean. And on behalf of Prytanean, it's my honor to introduce our homecoming lecturer and 2012 Faculty Award winner, Professor Diana Bautista. Professor Bautista received her bachelor degree in biology from the University of Oregon, her PhD in neuroscience from Stanford University, and was a postdoctoral fellow in physiology at the University of California, San Francisco. She joined the faculty at UC Berkeley in 2008. Dr. Bautista's lab studies the molecular and cellular mechanisms of itch, touch, and pain under normal and disease conditions. Her work has been recognized by numerous awards, including the Society of Neuroscience Young Investigator Award, a Howard Hughes Medical Institute Scholar Award, and an NIH Director's Transformative Research Award. Today, Professor Bautista presents How Do You Feel? Molecular Mechanisms of Itch, Touch, and Pain. And with that, Professor Bautista, please take it away.
Great, thank you so much, Tina, and thanks everyone for joining me today. I'm really excited to tell you about some of my lab's work. And so I'll just see if I could share my slides. And how is that looking? Everyone can see that. Great, thank you very much. All right, well, as Tina mentioned, my lab studies the mechanisms underlying itch, touch, and pain. And we know a lot about the senses. Let's see. So Aristotle was the first to define the five senses that allow us to interact with our world. And we know a lot about the senses. We know a lot about hearing and sight and smell and taste, but compared to the other senses, our sense of touch is the least understood. And to me, this is really surprising given how important touch is in our everyday lives. We know that touch is very important for emotional well-being. The bond between a parent and child is first forged through touch. Premature babies in incubators have regular scheduled periods of physical contact with caregivers and family members to ensure proper brain development. And I think during this pandemic, we've all felt the impact that loss of physical contact with friends and family members has had on our emotional well-being. Touch is also important for everyday tasks that we often take for granted, like walking in a straight line or tying our shoes, or more complex behaviors like learning to read Braille, or maybe something we do all the time now, like texting. Touch-sensitive neurons mediate our ability to do all of these tasks. And altered tactile sensitivity is the #1 complaint in the clinic. Examples include tingling and numbing associated with repetitive use injuries, like carpal tunnel syndrome, traumatic injury, diseases like cancer can lead to debilitating chronic pain, and chronic itch is associated with many disorders and is quite prevalent. Why do we have a sense of touch and pain and itch? We know that pain can be protective, even though we generally think about it in a negative way. It plays an important role as a warning system that triggers protective reflexes so that we don't constantly burn ourselves when we're cooking. We'll use a pot holder. It teaches us behaviors that are protective. Even inflammatory pain plays an important protective role. Again, here, if you've had a sunburn, you remember maybe next time to cover up or to bring sunscreen with you, or if you sprain your ankle, you take time. That pain prevents you from walking on it. That gives you time to heal. But chronic pain serves no purpose. It's not a warning system. It's not allowing you to heal. It persists for long periods of time in the absence of any disease or any injury. And there's a couple of great articles really showing how the loss of pain and chronic pain are equally debilitating. So this is a New York Times article called Painless that describes Ashlyn Blocker, who suffers from congenital inability to detect pain. When she was a child, she was constantly injuring herself, playing sports. One time she was cooking and she dropped the spoon in the boiling pot of pasta water she was boiling and reached right in and grabbed the spoon and didn't feel it at all. So she was constantly injuring herself, but never had any pain sensations associated with the severe tissue damage she experienced. At the other end of the spectrum, this is an article I'm highlighting about Cameron Bosdog, who suffers from complex regional pain syndrome. And in the absence of any injury, she experiences burning pain sensations over extended parts of her body. So loss of pain and persistent pain are both very debilitating conditions. And both of these conditions can be associated with other disorders, but pain is also a disease in and of itself. Chronic pain affects more than 10 million Americans annually, and no treatment cures or even significantly relieves suffering in most patients. And the challenge is to discover new genes that underlie chronic pain so that we could develop new therapies that target pain at its source, so that we could effectively cure pain, and to develop new treatments that don't suffer from severe addiction and tolerance that we see that's fueling the opioid crisis. So I'll just tell you a little bit about some basic science about our touch, pain, and itch system. So the somatosensory system is basically just clusters of neurons that are green in this cartoon that innervate extended parts of your body. Your skin, your viscera, all of your organs are innervated by these somatosensory neurons. And they detect environmental stimuli. So they mediate our ability to detect gentle pressure, as well as noxious pressure or pain. It gives us the ability to detect temperatures, as well as chemicals, both in our external environment. So for example, if you were cooking and you squeezed a lemon and got some lemon juice in your eye, these neurons get activated. But these neurons are also not only detecting environmental stimuli, they're constantly monitoring chemicals internal, such that if something goes awry, for example, a cancerous cell or tissue damage, then these neurons get activated as well and send out that signal, perhaps that discomfort you might feel that says, maybe you better go to the doctor. My lab studies somatosensory neurons from human donors, but we also use mouse as a model system. And here's just an image of a mouse embryo where all of the somatosensory neurons have been labeled with a green dye. And you could see that the general outline is very similar to that of the human. And we share many of the same genes and cellular signaling pathways that mediate these diverse sensations. So these somatosensory neurons, we have millions of them, and they roughly come in two flavors. There's a diverse array of them. So about half of your somatosensory system is dedicated to itch and pain. They're called nociceptors because they detect noxious things like pain. Oops. And the other half of your somatosensory system is dedicated to gentle touch that mediates behaviors like learning to read Braille, food texture preferences. I don't like mushy bananas for whatever reason, and that's mediated by mechanoreceptors. And of course, coordinated movements. And for you to experience itch or touch or pain, there's really 4 steps that happens in your nervous system. The first is called transduction. It's the conversion of a physical stimulus like heat or cold or pressure into an electrical signal, which is the currency of the nervous system. Then what happens is that electrical signal goes from the periphery to the spinal cord. And this is where a lot of processing of that information coming into your body occurs. You get modulation of that signal, and then that signal travels to the brain, and that's when you feel that ouch sensation when perception occurs. And so there's this transduction and transmission event. Those early events trigger protective reflexes, and then you have the perception of pain. So oftentimes what happens is you have that protective reflex, and then you feel the ouch sensation after that. So our goal is to look in the periphery to identify new pain targets that mediate transduction, that first step in that pain signaling pathway. So the big questions that we're trying to understand is how do innocuous and noxious stimuli excite these sensory neurons? What are those molecular players that trigger those that physical stimulus to become an electrical stimulus in your body? And also we want to understand how are pain thresholds set so that we could distinguish between the brush of a feather and the prick of a pin, that we know to avoid putting our hand in the fire or touching dry ice when we're working in the lab. And of course, we're interested in understanding not just what happens under normal conditions, but how does injury or disease lead to altered pain sensitivity? Are there different molecules? Are those molecules changed in some way? Or are there different cells and circuits that mediate those signals from the periphery to the brain? So what I'll do is tell you about 3 approaches that my lab has taken. And the first is to look at natural plant products that target our somatosensory system. So you've probably all eaten chili peppers and felt the heat, menthol and felt the cooling sensation, and perhaps that pungent experience from eating wasabi. By studying these chemical compounds that plants use to ward off predators, although humans, unlike other animals, have learned to like these types of experiences. Most mammals avoid these plants because they don't like these sensations. And by using these natural products, molecular mechanisms underlying heat and cold sensation, as well as mediators of inflammation triggered by the wasabi receptor, have been identified. And one example is that we used menthol to identify a gene called TRPM8 that encodes a menthol and cold sensor in somatosensory neurons. And so we could take these somatosensory neurons and see that they become activated in the presence of menthol or cold when this gene is expressed and functional. And this is just an experiment, an example of an experiment where we looked at the activity of cold and menthol sensitive cells. And what we see is that there's more activity, more electrical current that you see not at body temperature, but once the body, that part of the body, your skin touches ice and becomes cold. And the colder your skin becomes, the more electrical signaling you get. And then eventually that triggers those protective reflexes. We were able to confirm that this gene is important for cold sensation by generating a mouse that lacks this sensor, this gene. And so here's just an example of one of these experiments that we did. And this is an example where we took a regular wild type mouse. It has a functional copy of the tryptamate gene, and we gave it a choice between 20 degrees and 30 degrees C. There's a cold difference there, but it's not really noxious cold. But you could see very easily in this video that the mouse prefers to be on the warm side, and it's avoiding cold. And we hypothesize that that's being mediated by the TRPM8 channel. Now, if we look at this mouse's brother where we deleted TRPM8, what you can see is a very different behavior. This mouse now is no longer sensitive to a wide range of temperatures. In this example, it's innocuous cooling, but even really noxious cold temperatures. If we let the mouse, it would sit on a very icy cold plate indefinitely. It turns out that TRPM8 is not just mediating cold sensation in mice, but plays a key role in cold hypersensitivity associated with nerve damage and chronic migraine and cold hypersensitivity in humans. And it's associated with genetic variations in this gene are associated with a number of diseases that lead to chronic pain. And also it's right now, TRPM8 is a key drug target for treating pain associated with chemotherapy-induced peripheral neuropathy that causes severe nerve damage and hypersensitivity to cold that persists in about 60% of chemotherapy patients long after they stop the chemotherapy and their cancer is in remission. We're also studying animals that have unique tactile systems. So one example is the star-nosed mole. It's the most tactile sensitive animal that has been studied so far, and it lives underground in a tunnel system where there's a lot of other animals competing for food. And using this specialized tactile organ, it can detect small insects that no other animals can detect and has a competitive advantage in the ecosystem. So here's just a video where we dissected up little pieces of worm. This is an experiment done by my collaborator, Ken Catania. And what you can see is a normal mole that doesn't have a specialized star organ licks up a few of those pieces of worm, but basically walks right over them, even though he's hungry. Here's the star-nosed mole. And what you can see is it's very efficient at both detecting and eating these very small pieces of worm that students can't detect. So they're really small. And what this animal does is it reaches out and uses this specialized organ as an ultra-sensitive touch sensor, probing the surface, looking for food, touching it twice and eating it within the blink of an eye in 150 milliseconds. And so by studying this animal and by looking at the genes that are uniquely expressed in the touch sensitive neurons that innervate this touch organ. And I should mention that this touch organ is really small. It's about the size of your thumbnail. And even though this organ is so small, it's innervated 10 times more than the human hand. So by looking at these neurons and by looking at the molecules that are enriched in these neurons, we identified a key regulator of mechanical pain and pain hypersensitivity. And when we knock out this gene in a mouse and apply pressure to the paw, we found that they no longer display normal pain reflexes. So we could poke mice, and about 80% of the time, they don't respond at all compared to normal mice. And what we know is that this is a key regulator of touch, not just in moles and mice, but this gene is expressed in touch-sensitive pain neurons in humans as well. And we've been able to show that low levels of this gene cause mechanical hyposensitivity, loss of touch sensitivity that's indicative of diabetic neuropathy, where repetitive injury due to a lack of touch sensation can lead to injuries and in some cases amputation. That normal levels of activity of this gene are important for setting normal pain thresholds, and that under abnormally high levels of activity, it can lead to chronic pain and mechanical hypersensitivity. And finally, I'd just like to spend the last few minutes telling you a little bit about our most recent work, where we're looking at mechanisms of inflammatory disease, including chronic itch, asthma, and most recently, COVID-19. So you've all probably experienced an itch from a mosquito bite or an allergic rash, and perhaps found some creative way to scratch that itch. These are examples of acute itch, which is an irritating sensation that triggers a desire to scratch. And itch, like pain, can be very protective. It protects us against burrowing insects that carry diseases like malaria or Lyme disease by triggering scratching to not only remove the insect, but to trigger an inflammatory response that washes away infected cells. Itch is highly conserved. It's found not just in mammals, but fish can rub themselves on coral to get rid of pathogens as well. They can, eels, for example, can also visit cleaning stations where there are other fish that remove the pathogens. And even flies, Drosophila fruit flies, have an itch-like behavior to remove mites from their body that are capable of causing tissue damage or disease. And if you're interested, there's an incredible New Yorker article called The Itch about a patient who had neuropathic itch due to nerve damage and had a perceived itch that was so all-consuming that she actually scratched through her skull and gave herself brain damage. And I think it's an extreme example, but it highlights how the power of itch and the desire to scratch can be all-consuming. So unfortunately, 1 in 10 people will suffer from chronic itch at some point in their lifetime. Itch is associated with allergies and skin conditions like atopic dermatitis or eczema. Kidney and liver failure can lead to severe itch. And sometimes diabetes can lead to itch or pain, depending on the nature of the neuropathy. There are cancers like lymphoma that cause itch, infectious diseases, and there's a growing list of drugs that have chronic itch as a side effect. And it's thought that all of these different disorders cause itch through different mechanisms in different cells. But the one thing that they have in common is that antihistamines are completely ineffective at alleviating chronic itch. So there's a lot of interest in identifying molecular mechanisms of chronic itch with the idea that that might lead to a development of new drugs and therapies to treat chronic itch disorders. There's a subset of your somatosensory system that's dedicated to itch. And there's 2 classes of neurons. There are histamine-dependent neurons that become activated when, for example, you get a mosquito bite and you get histamine release and you get that itching and redness surrounding the bite. That's mediated by a special set of neurons that mediate histamine itch. And antihistamines are pretty effective, although not completely. Then there's this other part of the nervous system that mediates histamine-independent itch that is thought to be linked to chronic itch. And my lab has been really focused on that, understanding that pathway over the last 10 years. And we quickly realized that itch is chronic itch is a very complex disorder, and it involves the interactions between three cell types. The sensory neurons that mediate itch sensations, the skin cells themselves, keratinocytes, that signal that to sensory neurons, and also to immune cells. And what we see looking at disease skin is that all three of these cell types show hyperactivity and abnormal interactions. And so we set out to define the cells and the signaling molecules that drive chronic itch and inflammation. And we decided to start by tackling atopic dermatitis or eczema because it's the most common chronic itch disorder. About 20% of children have eczema and about 10% of adults worldwide. And it's characterized by these really dry, itchy patches of red skin. But the main complaint from patients is that all-consuming desire to scratch. And in this case, scratching only partially alleviates the itch and actually causes more damage in a vicious itch scratch cycle that causes more tissue damage and then more itch sensations. The other reason we were really interested in studying atopic dermatitis is because it's an entryway disease to other disorders. About 70% of kids with severe eczema go on to develop other atopic diseases later in life, including food allergies, allergic rhinitis, and a severe form of asthma that's really difficult to treat. And atopic diseases were traditionally considered diseases of epithelial cells and immune system. But what we know now is that peripheral neurons, those neurons of the somatosensory system, interactions of those neurons with our immune system together drive skin inflammation. And this has been the work of two students who graduated recently, Rose Hill and Carolyn Walsh, who decided to tackle this problem. What they found was that there was a unique subset of interactions between skin cells, neutrophils, and a special new class of sensory neurons that we've been looking at that drives the development of itchy lesions. And as this moves from an acute to a chronic itch disease with persistent itch, that there is a second class of neurons as well as other immune cells that then drive the chronic disease state. And I won't go through the data, but we identified several signaling molecules and new cell types that we think contribute to this development from an acute itch to a chronic disease. We're very interested in defining molecular mechanisms, neuroimmune mechanisms in skin inflammation. And actually what we saw was that this change in the skin drove massive changes across the entire nervous system and saw a wave of inflammation starting in the skin to the cell bodies becoming hyperactive, and looking at the spinal cord as well, that there were these dramatic changes in how those electrical signals in the nervous system process. And what we're doing now is trying to understand how this wave of inflammation in the nervous system and how immune cell infiltration at multiple levels contributes to this atopic march from inflammation in the skin to inflammation in the lungs to trigger asthma later in life. And the hypothesis that we're currently testing is that these sensory neurons are playing a key role in that transition, and that the nervous system is playing a key role in disease. We already know that the airways are densely innervated by these somatosensory neurons, and that they are closely interacting with lung epithelial cells in the airway. And a lot of the same molecules that we've identified in the skin are abundantly expressed in neurons and epithelial cells in the lung. And now we're looking at the role of these molecules we've identified in eczema in the transition from homeostasis and normal airway function to a state of inflammation that could lead to respiratory distress. And of course, this got us very interested during the pandemic in looking at these neuroimmune interactions in SARS-CoV-2 mediated airway inflammation, because these same sensory neurons that are regulating airway inflammation and airway function under normal and disease states are also infected by SARS-CoV-2. And so overall, hopefully, one of the takeaways from today's lecture is that by studying really basic science questions, we can discover new gene targets that might lead to the discovery of novel therapeutics for treating human diseases. And also, I hope, I just want to thank the people who did the work in my lab. All of this work was done by trainees, so graduate students, postdocs, and undergraduates. I've had over 30 undergraduates in my lab contributing to research over the last 14 years. And none of this would have been possible without their hard work and dedication. And I'd just like to thank the National Institute of Health that has been a major funder of this basic research, with the idea that by supporting basic research that this might lead to new discoveries in medicine and therapeutics, as well as the Howard Hughes Medical Institute, who funded my research as well. And with that, I'd be very happy to take any questions that you might have. Thanks.
Well, thank you, Professor Bautista, so much for your lecture. Super fascinated by and lots to unpack from your research and really the connections between our nervous system, sensories, temperature, and mechanisms around itch and inflammation. So I will say I'm definitely the mouse on the right who prefers the warmer temperatures, and I love to scratch. So I'm really curious, kind of, are there questions from the audience? Let's take a look at that. We have one. Okay, so first question to you, Professor Bautista. Is there already medicine approved based on this research to help us with chronic itching and pain?
Oh yeah, that's a great question. Not yet. So most of the treatments out there are targeting the immune system. And we think that drugs that target both the immune system and the nervous system might be more effective. So we'll see. A couple of the new drugs that have been recently developed and are out there are molecules that target immune cell signaling molecules. And some of those we think, and other people think, act on the nervous system as well. So some of the latest drugs that block interleukin receptors might be really effective. And we're really excited to see where some of those new drugs, how they do outside of the clinical trial now that they're becoming somewhat more available to eczema patients out there.
Yeah, actually, that makes me think about the Dupixent shots. I keep seeing those commercials for it, and whether or not it really lives up the hype or it actually works. So,
yeah.
Okay, we have some more questions. Wow, a lot more questions coming in. Okay, next question. If you have eczema, what is the likelihood of developing asthma?
Yeah, so so people with severe eczema, people sometimes just have a little patch or some seasonal eczema. People who have persistent eczema as a child, that's more on the severe side, where they had to receive medical treatment, were taking prescription steroids for extended periods of time. About 60 to 70% go on to develop asthma. So it's quite high. If you think about starting with 20% of children, that's a lot. And we don't know why, but the cases of atopic dermatitis and eczema and food allergies are really on the rise, especially in Japan and Sweden and the United States. And so finding new therapies as these numbers are growing, I think is really important.
Absolutely. We actually have there was another question kind of related to dermatitis. So do all asthma patients have atopic dermatitis or just a small percentage?
Yeah, so flipping it, many asthma patients have atopic dermatitis, but many do not. And so it seems like if you have eczema, if you have eczema, you're much more likely to get asthma than the general population. But there are people with asthma who didn't have eczema. And the people who had eczema often lose it and then they have asthma. So I have asthma. I had eczema as a kid. I don't have eczema anymore, but.
Oh, wow. More questions coming in. Okay, next question. How likely is someone to develop an inflammatory lung disease, ILD, if they experience severe eczema in their childhood?
Yeah, that's a good question. It's not clear. I think that having severe eczema does seem to prime your system. There's been a lot of studies where even just looking at allergies, that if you had eczema and you now are exposed to an allergen, that you have a much more exaggerated response. And so, but specifically for inflammatory lung disease, and I think we're all thinking about this in the context of COVID-19. Are we more likely to have a severe case if we're infected? I don't know, but it's good to be extra careful, especially if you have exaggerated immune responses in the past.
Absolutely. Next question is, can you say anything about chronic pain from inflammatory muscular diseases such as polymyalgia rheumatica?
Yeah. So right now, one lead drug candidate for like rheumatoid arthritis and other inflammatory pain, thinking about joints, muscle inflammation, is the TRPA1 gene, which encodes the wasabi receptor. So when you eat wasabi and you feel that really transient, pungent sensation, that's quite different from eating chili peppers, that activates this inflammatory receptor. Of course, with wasabi, it's just low levels, but it turns out that plants in the Brassica family target this inflammatory pathway to ward off predators from eating them and destroying their seeds. But inside, and it turns out that those wasabi and other compounds in those types of plants tap into this receptor that normally responds to extreme buildup of natural inflammatory mediators inside our body. So under inflammatory conditions, this TRPA1 molecule becomes very activated and is thought to trigger pain associated with inflammation. And so there's been some promising clinical trials, and we'll see what happens next.
Next question is, why do the eczema areas appear to be on the face, my breast, inner elbows? And why is it what's so special about those areas?
Yeah, so there are these hot spots, and one theory that there's some evidence is that the microbiome plays a big role in driving eczema. And there's been a lot of studies, especially in children, that the bacteria in these atopic lesions are quite different from the normal healthy bacteria. that we have in on our skin. And those hot spots also, even in healthy people, tend to have different types of bacteria there, where you know, there's like where our skin is coming into contact, and you know, places where we have limb bending, elbows, little spots where the microbiome, even in healthy people, are different, seems to be these hot spots for eczema to develop. And the idea is that maybe those areas then have a different type of microbe that's making these hyperactive responses and contributing or initiating. Exactly how they fit in to the disease pathogenesis isn't really known, but that's a theory right now.
Very fascinating. Next question. I didn't ask this question, but I'm very curious myself. Is itch tied to stress?
Oh, yes. Oh my gosh. It so is, right? And so, and this is true for itch, and it's true for pain. How our body, you know, our mental state, right? That brain body connection that we hear about, stomach inflammation. There's definitely a connection with stress hormones, right? That can impact and travel throughout the body. And so definitely asthma, chronic itch, chronic pain are all triggered by and can be modulated by stress hormones.
So Professor Bautista, another question came in. This one is actually, we're all looking for the solve, right? Like how to just have our troubles go away. So one of the questions asked, since genes are identified, is it possible to do gene editing in the future to prevent atopic dermatitis, for instance?
Anything's possible. I'd say we've made amazing advances here at Berkeley in gene editing and CRISPR-Cas9 technology from UC Berkeley. But I think we have a long ways to go before we start editing ourselves. And I think with, you know, thinking about atopic dermatitis, you know, even some of the drugs that have been developed have off-target effects. And so what we still need to know a lot more about these disease genes. How specific are they? We don't want to start editing things that play other important roles. So for example, the capsaicin receptor was a very promising drug target for treating heat pain hypersensitivity. So like Cameron Bosdog, who suffers from complex regional pain syndrome, where you know, if she walks fast and gets a little warm, then starts to feel burning pain sensations. But at the same time, I mentioned pain is protective. So if you turn off some pain genes, you might then lose these protective behaviors. You might change temperature regulation. So it is complex, as in all disorders and diseases.
So this is going back, this question, very fascinating also. So someone said, this is the first time I've heard of plants seeking to protect themselves against being eaten. So do all plants try to avoid being eaten? And that was the original question. So my follow-up to that is, I'm wondering if that's the reason why I developed a rash actually from gardening and trimming my tomato plants.
Oh, yeah. Yeah, this is super interesting. So it depends on the plant. And so one kind of cool story is about the capsaicin plant, that the chili pepper plant that produces capsaicin. And so squirrels and deer, when they, and humans, right, our digestive system destroys those seeds such that they can't get propagated. But it turns out that birds have a different digestive system. They could eat the plants. The seeds pass through their digestive tract intact and they fly away, and that promotes plant spreading. And it turns out that both humans and mice and birds all have a capsaicin receptor but the birds don't have the binding site for capsaicin. So the bird receptor is activated by noxious heat, but is not activated by capsaicin. So it's thought that in this case, these plants evolved with animals to promote bird eating, being eaten by birds, which disseminate the seeds, that there's been this coevolution, but at the same time preventing mammals from eating them. So it is quite interesting.
Very fascinating. Also new perspective on plants too. Next question, Professor. Okay, so I always see these on Instagram. There's even a few doctors that do TikToks on this. So does retinol help treat eczema? Burning question.
Yeah, I think some people claim it does and some people claim it doesn't. So I would say it's worth trying and to see for yourself. But yeah.
Okay, next question. Lots of questions here. Does the spinal cord actually process electrical signals, or do they just pass them to the brain for processing?
Oh yeah, that's a good question. So there is a lot of processing that occurs at the level of the spinal cord, and several drugs, pain drugs and opiates, right, can target both pain signals at the level of the brain, but also at the spinal cord. So there's a lot of processing there, and it's thought that that might be a much better place to target pain. And so there's a lot of interest and a lot of people studying spinal cord modulation and finding targets there.
Next question is, why are hospice patients asked and tested if they feel pain at or near the end of life?
Yeah, so as we age, our tactile system changes quite a bit, and we can become less sensitive to gentle touch. We could have trouble with touch receptors and coordinated movements. And in some cases, there's a heightened experience to pain. At the same time, in hospice, oftentimes our senior patients might have trouble communicating or are not as communicative. And so they're often asked that repeatedly to make sure that they're feeling okay, that they're accurately describing what types of sensations, and to make sure that medications, right? We don't have any good way to measure how effective medications are. And so right now, and this is part of the problem, right? So right now it's just based on asking, is this dose helping? Do you need more? And just ensuring sort of proper hospice care.
Thank you. So we have time for one final question, and it just happens that someone just released a question to us. So last question, can the pain touch sensors that may be damaged by disease or by chemotherapy regain their functions?
Yeah, that's a great question. So if you look at chemotherapy-induced peripheral neuropathy, about 60% of patients develop it. And so we're very interested in understanding and looking at genetics to ask what makes one individual resistant and one individual develop this peripheral neuropathy. And then of those patients who develop peripheral neuropathy, about 60% recover after the chemotherapy stops, but those other patients have nerve damage that doesn't reverse, and have persistent pain and pain syndrome for the rest of their lives, even after the cancer and the chemotherapy is gone. And so the question that many people are trying to understand is, what can we do for those people? we're trying to prevent it from happening in the first place, but are there factors that can be made to help regenerate or regrow those damaged neurons or repair their health? And that's a very active area of research.
Well, thank you, Professor Bautista. Really such a wonderful, enlightening lecture. I definitely learned a lot more about really processing what itch means, all the sensories, and even now I understand why my tomato plants are the way they are. And I'm the mouse of the right in the study. Warm temperatures, everyone. Amazing lecture. Thank you so much for building our questions. This concludes Q&A. We had like 13 questions. Amazing. For all of you joining us virtually, happy Friday, and happy Friday, Professor Bautista. I hope that you'll all enjoy all the festivities to come. And with that, we're going to take it away, the closure.
Thank you. Bye.